Welcome to Grow Bio channel. In today's video, we're going to see about BED 2008 question papers solved with explanation. First is virus-mediated transfer of cellular genetic material from one bacterial cell to another by means of virus is called as. Options are transduction, transposition, transformation and transfection. The answer is transduction. It is the transfer of foreign DNA introduced by the virus into the E. coli or some kind of bacteria. So this is known as horizontal gene transfer. Here you can see T2 phage which infects the bacteria. Here the DNA only introduced into the E. coli and then it produces proteins and replicates its DNA and then assembles and then it produces the progeny of the phage. This process is known as transduction. So other options are transposition. It means position is changed by jumping genes, which is also known as introns. So in some bacteria, the jumping genes jumps and creates the mutation. This is known as transposition. Next is transformation. That is transformation of one strain to other strain in the same species. That is, in Griffith experiment we have seen like live R2 is changed to live S3 by taking the S3 gene that is present in the media. So this experiment is example of transformation. And next is transfection. This is the genetic material is deliberately introduced. So here two bacteria are involved. In these three, transposition, transformation and transfection, no virus is involved. So the answer is transduction. Next, plasmid cloning vector PBR322 contains ampicillin resistant and tetracycline resistant gene that confer resistance to both ampicillin and tetracycline respectively. So this PBR322, it is itself the transformed E. coli. It has resistance to ampicillin and tetracycline, means it grows in the media which contains ampicillin and tetracycline too. So the tetracycline resistant gene contains site for the restriction endonuclease BAMH1. Not only BAMH1, other thing like SAL etc. is present. So similarly for ampicillin resistant P57R and PVUL restriction endonuclease site is present. So if there is cut in that site, then this resistant gene will lose its capability to survive in the tetracycline media or ampicillin media. So here what happens, the tetracycline gene contains the site for endonuclease BAMH1 and it is cleaved by BAMH1. So then other BAMH1 restriction fragment from a different DNA molecule is joined by using the DNA ligase. This means the site is cut and other fragment is introduced and it is ligated. So when it is cut, then tetracycline resistance will be lost by the new transformed plasmid. Okay, so and it is used to transform the E. coli cell. Under these conditions, which of the following statement is true? The answer is B. Tetracycline can then be used to select the transformed E. coli carrying the non-recombinant plasmid. The PBR322 it itself a transformed E. coli plasmid. So in presence of tetracycline, only the tetracycline resistant containing plasmid can only be grown. So here you can see a comicon which has two tetracycline resistant and ampicillin resistance gene with it. So here the transformed one, the recombinant one, here the BAMH1 cuts it, so tetracycline resistance is lost. So this recombinant one cannot survive in tetracycline containing media because it lost its ability to survive there. Only the non-recombinant plasmid can be grown in that media. So the answer is B. Next, which subunit of E. coli RNA polymerase is responsible for gene selection? Options are alpha, beta, omega and sigma. Answer is sigma. The prokaryotic RNA polymerase is made up of 6 subunits. Okay, the 5 subunit is common for all the gene. The 6th subunit, it is the sigma subunit which is responsible for gene selection. So the sigma subunit is the functional subunit. It differs for one gene to another. So which gene must be transcribed must be known by only this sigma subunit. So the main important functional subunit is sigma subunit. So the answer is obviously sigma subunit. So next is, which one of the following radioisotope does not emit beta rays? So here, what is beta rays? Beta rays is produced on radioactive decay of atomic nucleus. Okay, it can be both electron or it can be positron. So that molecule will be decayed by emitting the beta rays. So here the options is 14 carbon tritone, that is 3H, and 32 phosphate, 125 iodine. The answer is 125 iodine, which do not emit the beta rays because it is stable till 60 days. Okay, it is also used in therapeutic purpose. So the answer is 125I. Other 3 is unstable. Okay, it emits beta rays. Next is, which of the statement about tRNA is false? Now we can see the options. All organisms have more than 20 tRNA genes. It is true because nearly 22 tRNA can be present in any organism. We have 22 amino acids, so anticodons will be present in tRNA. So next is, the three-dimensional structure of tRNA looks like clover leaf. It is false because the two-dimensional structure of tRNA looks like clover leaf, not the 3D structure, because it can't be logged into ribosome in clover leaf structure. The three-dimensional structure is different for tRNA. Okay, so the second option, the 3D structure of tRNA looks like clover leaf, is false statement. So the answer is B. However, we can see other options too. tRNA contains modified bases. True, it contains modified bases so that anticodons will be there. So amino acid can be joined to other amino acid and forms peptide bonds. So C is true. And next is, the sequence of last 3 nucleotides at the 3' end of all tRNA is the same. True, because last 3 nucleotides will be obviously be stop codons. So D is true. So here the answer is, the 3D structure of tRNA looks like clover leaf is false. Okay. Next is, collagen consists of 3 helical chain. Yes, it is triple helix structure. So it contains glycine and proline amino acids in each chain. True. So for every 2 amino acids, the third one will be glycine. Okay. So first 2 can be anything, like any amino acids. Okay. The overall structure of each polypeptide in the collagen molecule is options is given. The answer is polyproline 2. Because as I said, for every 2 amino acid, third one will be glycine. But the first 2 can be proline or hydroxyproline. Okay, that too mainly polyproline 2. So the answer is polyproline. Okay. Next is, in NMR spectroscopy, the absorption spectra result by the absorption of one of the following electromagnetic radiation by spinning of nucleus. For this we have to know about NMR. NMR focus on only nucleus and not the electrons. So the nucleus will be spinned and electromagnetic radiation will be exposed. So the EMR produced by that particle is measured by NMR. So what EMR is produced? The options are given: UV, infrared, radio waves and microwaves. The answer is radio wave. Okay. The absorption of electromagnetic radiation in the form of radioactive waves. Frequency from the region of 4 to 900 megahertz by NMR. It is produced by the atomic nucleus. Okay. By absorbing that radiation, it will be producing the result. So the answer is radio waves. Next, we can see the ribonuclease solution gave an absorbance of 1 at 278 nanometer in the UV spec using a 1 centimeter quartz cuvette. Given that the molecular extinction coefficient of the enzyme at 278 nanometer is 10 square molar per centimeter, the concentration of the enzyme would be. 4 options are given. So in order to know the concentration of enzyme in UV spec, there is a formula. The answer is 10 mm. There is the formula. It is got by Beer-Lambert's law. The log W equal to I naught by I, which is equal to epsilon into L into C. Okay, the epsilon means molar absorptivity, and L is length of the solution that light must pass through in centimeter. Okay, the length must be taken in centimeter, how many length that the light must be passing through it. And C is concentration of solution. So here I naught by I can be written as absorbance. So absorbance equal to epsilon into L into C. Absorbance here is 1 at 278 nanometer. So A is 1 equal to epsilon is molar absorptivity, that is 10 square per molar per centimeter. So 10 square is taken as epsilon, and length of the cuvette is 1 centimeter. So for that 1 centimeter only the light must pass. So L is 1, and we have to know the concentration. So 1 by 100 equal to C. So C is 0.01 molar. In millimolar it can be written as 10 millimolar. Okay, 1 molar is 1000 millimolar. Similarly 100 millimolar is 0.1 molar, and 10 millimolar is 0.01 molar. So the answer is 10 millimolar. Next question. Two proteins have the same molecular mass as well as the same isoelectric point. The best way to separate them would be to use. Answer is reverse phase chromatography. So based on the hydrophobicity of the particle, the reverse phase chromatography is done. Here the stationary phase is alkyl chain. So hydrophobic particles will be grabbed by that. Hydrophilic particle will be moved through the column and come out of the column. So the point here is it is not depend on the molecular mass or isoelectric point. It depends on the hydrophobicity of the particle. And next three, the gel filtration chromatography, it is based on size and molecular weight. Okay, and ion exchange chromatography is based on ion, and chromatofocusing means it is based on isoelectric point, that is affinity to the ion exchanger. Okay, so these three depends on the isoelectric point and molecular mass. So these three is wrong. The answer is reverse phase chromatography, which is based on hydrophobicity. Next is one strand of double stranded DNA is mutated, changing all cytosine to uracil. After one round of replication of the mutated DNA strand, the melting temperature of the resulting DNA will be. Obviously the answer is lower, because uracil in DNA, see, DNA is made up of adenine, thymine, guanine and cytosine, and not by uracil. Okay, the uracil is present only in the RNA. So changing all cytosine to uracil, what happens? The GC bond will not be there. Okay, because G only binds with C by 3 hydrogen bond. So all GC bond will be erupted. So what happens? If there is low GC bond, then the DNA will be unstable. So the melting temperature will be low. At lower temperature itself, the DNA will be melted. So answer is lower. Okay. So next is, which type of restriction enzyme does not require ATP? The answer is type 2. It need only magnesium 2 plus for cleaving the site in close proximity or cleaving at the site itself. The other three is different. The type 1, it will cleave at random site far from the recognition site. So type 1 needs ATP, but it will cleave far. Type 3, it cleaves only the palindromic sequence. Okay. Type 4, it cleaves only the methylated DNA. Okay. Only the type 2 doesn't need ATP, it only need Mg2 plus to cut in the close proximity. So the answer is B. Next is, southern blotting technique is used for. The answer is the detection of DNA fragments on the membrane by radioactive DNA probe. We know southern blotting is for DNA and northern blotting is for RNA, western blotting is for protein. So the option A, C is wrong because it is based on RNA and protein. The B and D is based on DNA fragments only, but it is B is by radioactive probe, D is by radioactive antibodies. So here radioactive probe only used in blotting technique, not the antibodies. So the answer is B. Next is, which of the following is not found in E. coli replication fork? These four options is in need for replication. DNA A is the first one to approach ORAC. So it is responsible for unwinding the DNA, and after unwinding it, it will be removed. So it will not be present in replication fork. Okay, it is responsible for replication and it will not be present in replication fork. So the answer is DNA A. Okay, it is found only in the initiation of replication and then cleaved by polymerase and DNA B. Okay, so it will not be present after the initiation, thus not in fork 2. Next is primase. Primase is a need for adding the primer so that the lagging and leading strand will be there. So in leading strand one primer will be needed and lagging strand more primers will be needed. So the primase is necessary in replication fork. pCNA is proliferating cell nuclear antigen. So it is present in prokaryotic and eukaryotic and RK replication too. So pCNA is also present, and SSB protein, it is responsible for stagging the unwinding DNA so that it will not coil. Okay, so SSB protein is also responsible. So the answer is DNA A, which is not present in replication fork, but it is responsible for initiation. Okay, next is, which of the following is changing the fastest over the evolutionary time? The answer is microsatellite. It is present in exons. Okay, that is the coding part of gene, and change in the coding part mutates the DNA itself. So others such as order of genes, okay, on chromosome and DNA sequence doesn't change such easily, okay, because of proofreading activity. And change in introns doesn't change the coding part, so it does not leads to evolutionary itself, and there will be only movement. Okay, so it is known as jumping gene. So the option A, amount of intergenic DNA, that is introns, and order of genes on chromosome will not be changed such easily, and exon DNA sequence cannot be changed just like that because of proofreading activity. So the answer is microsatellite, which will be changing the fastest over the evolutionary time, which is present in the exons itself. Okay, the answer is C. Next is superoxide dismutase is an important enzyme for the maintenance of RBC and is defective in some neurodegenerative diseases. Which does this enzyme do? So the answer is A, catalyzes the conversion of O2 minus to H2O2 and oxygen. The SOD converts the O2 minus, okay, from the mitochondria P450 oxidases to peroxide and oxygen, that is hydrogen peroxide and oxygen, and then it changes it into water. This SOD is there to maintain the oxidative stress in the blood. Okay, so it is present in some neurodegenerative diseases such as Parkinson's, Alzheimer's, etc. So the answer is SOD catalyzes the conversion of O2 to H2O2 and O2, and then it changes it into water. Okay, next is, which of the condition would not promote denaturation of double-stranded DNA? Okay, first is heating to 100 degrees Celsius. We all know PCR denaturation temperature is 94 to 98. So it will denature the DNA at 98 degree itself. So 100 degree is obviously it promotes the denaturation of double-stranded DNA. So A is wrong. And next is adding high concentration of sodium citrate. Sodium citrate is used to store the DNA, double-stranded DNA. Okay. It also stabilizes the DNA structure to some extent and enhance the melting temperature. Though, see, salt ion charge reduce electrostatic repulsion between the phosphate charge because 3-phosphate is present consecutively, as we all know about the DNA structure. So this reduces the electrostatic repulsion between the phosphate too. So adding high concentration of sodium citrate doesn't affect the denaturation at all. Okay, so the answer is B. Okay, adding high concentration of sodium citrate. However, we can see about other option too. Decreasing the ionic strength of the solution. So decreasing ionic strength means the ionic strength determines the pH. So the DNA denatures. Okay, when the pH is low, that is it is acidic to the nucleus. So DNA denatures. So next is SSB. SSB, that is single strand protein, is responsible for replication, repair and recombination. It separates DNA, which makes the DNA unstable, okay, which makes it for denaturation. So SSB is also responsible for denaturation. So the answer is high concentration of sodium citrate will not affect the dsDNA. Okay. Thank you. If you have any doubt, let me know by commenting in the comment section below. Subscribe and please press the bell button below. Thank you.